Okay, I think we can start now. So hello everyone. My name is Emily Delgouffe and I would like to welcome you to this special webinar. It's the very first joint edition of NYRA and ORCHID-NET. We are happy to have you all here today as we combine forces to explore the exciting future of fertility preservation and restoration in men. Before we get started, we will briefly introduce both networks and also our speakers of the day. So I will start by introducing the Network for Young Researchers in Andrology, better known as NYRA. So NYRA is an international network that supports young scientists in testis research by encouraging collaboration and knowledge exchange across borders. So we aim to provide a more informal platform for networking and discussion compared to the traditional congresses. So our network was founded in 2006, as you can see on our timeline here. And it was founded during the 14th European Testis Workshop by a group of young scientists, of some of whom you might still recognize today. And as you can see, over the years, NYRA has gone through a few different names, some definitely easier to pronounce than others, but in the end, we happily settled on NYRA. Another milestone was quite recently when we joined forces with the European Academy of Andrology, or the EAA, and we became their young arm, and this really strengthened our connection to the broader andrology community. So here is a nice overview of our current board. So we're a close team of young international scientists, and as you can see, we aim to include as many nationalities as possible to truly bring a global perspective to the andrology field. As I said before, at NYRA, we're all about bringing young researchers together. So to do that, we have a list of initiatives that we try to do all the time. And the first and probably the most exciting thing that we do each year is that we have a NYRA meeting. And this meeting is a great opportunity for young researchers to network, to share ideas, and most importantly, to give them a relaxed and supportive space to present and discuss their work. We also recently launched our very first NYRA school. This was together with the EAA and Andronet, and the focus was on omics analysis. So this was a great mix of hands-on learning, but also an opportunity to connect with experts in the field, but also peers. The third main activity of NYRA are the Andro Connect webinars, which we kicked off last year and is still ongoing now. So these are all free webinars that we are hosting. And in these webinars, we're discussing different career paths, and we're also sharing insights from people who are working within the field of andrology. We're also present at major conferences where we sometimes can host dedicated NYRA sessions or pre-courses. And here we try to make sure that also the young voices are being heard within these congresses. Last but not least, we're sending out seasonal newsletters, and we also post press highlights on our website. And we also are very active on our social media channels. So here we try to share NYRA updates, possible job opportunities, interesting events, and many more things. So if you would like to stay connected with us, make sure to check out our social media channels, which are linked here. So we are present on LinkedIn, X, and Blue Sky at the moment. We also have a website which you can access through the QR code here or via this website at the bottom. When you go through our website, you can easily subscribe to our newsletter via the subscribe button that's indicated here. And if you're looking to get even more involved, we also offer a NYRA EAA membership, which comes with extra benefits for young researchers. So if you're interested in that, you can click the other button that's highlighted here, and then you can find more info on this membership. So with that being said, I'm happy to pass the mic to Callista, who will tell you more about our partner in today's webinar, which is ORCHID-NET.
Hey, thank you, Emily, for this nice introduction on NYRA. My name is Callista Mulder. I'm based at Amsterdam UMC, but today I will represent ORCHID-NET, which is a very nice international network to improve fertility preservation and restoration for young males. Next slide, please. So in 2022, the ORCHID-NET network was founded, and we had the common goal to improve the quality of life for young males at risk of losing their fertility. And these are, for example, young boys that have cancer or benign hematological disease that are at risk of losing this reproductive potential, and to give them a chance of biological parenthood later in life. In the clinic, this typically goes through a testicular biopsy prior to their treatment. And within this group of experts, we noticed that there were quite some differences in care pathways for male fertility preservation between the institutes that were involved in our network. And we aim to improve this as a group of people. And we named ourselves ORCHID-NET, not only because we love the flower, but also because orchid actually means testis in Latin. And if you look at the roots of the flower, you can also see the resemblance to the human organ. Next slide. So we had some goals. We aim to enhance collaboration between experts both on the clinical side and on the research side for male fertility preservation and restoration. We also aim to combine our clinical data sets, to share our resources, both intellectually and practically, and to develop shared research projects, ultimately to provide benefit to our patients. Next. So since we are a international group, we usually meet online via Zoom or via Teams. And occasionally we get to meet in person, for example, at the European Testis Workshop or at the ECA last year in Stockholm. And in this, we are led by Professor Dr. Rod Mitchell from Edinburgh, who is currently the coordinator of this network. He is potentially eavesdropping from Athens as he was traveling today. Next slide. So currently we have 58 members in 19 separate countries, and we aim to be able to represent the countries that do fertility preservation at this moment, but also who aim to set up fertility preservation for young males in their country or their city. Next, please. So since we noticed that there were quite some differences when we talked to each other regarding fertility preservation, we started off with a survey where we tried to review what we were doing in the clinic regarding fertility preservation. And with this survey, we were able to identify over 3,000 patients within our institutes that had a testicular biopsy cryopreserved before the age of 18. And these were both for malignant diagnosis, but also for non-malignant diagnosis in 40% of cases, for example, benign hematological diseases, but also, for example, in case of transgender treatment. Next slide. And there were quite some differences in how the tissue is handled, both in, for example, fragment size, the cryoprotectant use, and the media additions that people were doing. Next slide. So this sparked the idea to write good practice recommendations on this matter. And we joined forces with ASHRA And together with a group of experts, we wrote this good practice recommendation. And we also got tremendous help from ESRI, especially Natalie Leclef, who helped us do this extensive literature research and guide us through the expert discussions to actually achieve this. Next slide. And these good practice recommendations are out now. So if you scan the QR code, you can find them. We have identified 44 key recommendations for fertility preservation involving testicular tissue cryopreservation in children and adolescents who require a gonotoxic treatment but cannot produce sperm. And these 44 recommendations, they revolve around the program setup, so what you might need to set up a program like this in your institute, about cryopreservation. Eligibility was also a big topic in this, so which patients should you offer this biopsy? And this, of course, goes hand in hand with counseling and suggestions for further research. Next slide. And with this, we're also looking at the future, because of course, in the end, we want to aim to help these young males have their families later on in life. So I will not spoil too much because this is, of course, the topic of Kyle's presentation. But we were quite proud to have written an expert review on whether the time is right to actually transplant immature testicular tissue to restore fertility in these, for example, childhood cancer survivors. Next slide. So this is what I'd like you to leave, you with. If you feel left out, we always welcome new members of collaboration. You can scan the QR code and then you go to our website and there's a contact form. Just reach out to us whether you have questions, you want to join us. And yeah, happy to collaborate. And I'll give the word back to Emily now.
Yes. So then it's my pleasure to welcome our two speakers of the day. They're coming all the way from the United States for today's webinar. So first of all, we have Professor Dr. Kyle Orwig, who is a professor at the University of Pittsburgh and the founding director of the Fertility Preservation Program at the University of Pittsburgh Medical Center and the McGee Center for Reproduction and Transplantation. He's a world leader in stem cell biology, and his work focuses on helping patients to preserve and restore their fertility. And today he will share both his latest scientific advances and also his personal experiences that shaped his career. Joining him today is Dr. Amanda Zielen, who is part of the same fertility preservation program where she works as a research manager. And she will present her recent results on searching for sperm in testicular tissues from peripubertal patients, providing insights into an important and also innovative area of fertility research. Before we dive into the presentations, I wanted to share a few quick notes. So this webinar is being recorded, and the recording will be made available after the webinar. And then maybe most importantly, we encourage all of you to submit your questions at any time during the webinar using the Q&A tool, which you should be able to see at the bottom of your screen. And we'll go to the questions at the end of this webinar during our Q&A session. So please, if you have a question, mention to which speaker your question is. Because this will help everything to run smoothly at the end and make sure that your questions get answered by the right person. So that being said, I will now stop sharing my screen and I will give the floor to Professor Orwig.
Okay, is everybody able to see my screen okay?
Yes.
Okay. Well, first of all, I want to thank Emily and Callista for organizing this webinar, and I'm sure Amanda, are honored to be invited to be the presenters today. So I will initiate the discussion. I've been asked to talk about both my career development and also some of our work, and then I'll end up by describing our center where we provide fertility preservation services. This is both a research and a clinical center, and so I'll pass the baton there to Amanda so she can tell you about some of the research that we're doing with our patients. So I want to start out by describing my career path, and I think the things that I want to emphasize are both the mentors who have been influential to me, but also the places where this has happened and what I learned there that I could take with me in my career journey. So I did my PhD studies at Oregon State University. At that time, I was studying ovarian function, and this was in farm animals, cows and sheep and pigs. And at that time, probably the expertise that I was learning was reproductive endocrinology. So this is where I learned the hypothalamic pituitary gonadal axis and specifically about ovarian function. From there, I went and did a postdoc with Mike Soares at Kansas University Medical Center. And there I learned about placental functions in rodents, but most importantly, what I learned there is molecular genetics, how to make transgenic animals and how to do gene bashing type of things, because I've always been interested in genes and their functions. I never really felt like an expert at placenta, so when it was time to go looking for a faculty position, I didn't feel like I could be the doctor of the philosophy of placenta. And an opportunity opened up to work with Ralph Brinster, who is a pioneer in producing transgenic and knockout animals. And this is something that I always have been excited about because those were new technologies back in those days. Interesting, he wasn't making transgenic animals anymore at that time, but rather he had begun studying the stem cells in the testes that make sperm, and he pioneered the method for transplanting stem cells from a fertile mouse into the testes of infertile mice where they could regenerate spermatogenesis and in some cases fertility. And I'll tell you a little bit about that. So during that time, I transitioned from a senior postdoctoral fellow to a junior faculty. That allowed me to write grants, and I got my first R01 grant when I was there, which opened the door for my opportunity to take my first tenure track faculty position. That's here at the University of Pittsburgh, and I've been here ever since 2003. I was recruited by Jerry Shatton, who was really a pioneer in everything related to assisted reproduction in monkeys and producing transgenic monkeys. And so I'll show you some of the monkey research, which I would never have been able to do without getting foundational education from Jerry and his group. Um, and finally, somebody who wasn't a direct mentor of mine, but was a career mentor of mine is Teresa Woodruff. She's currently at Michigan State, but when I met her, she was at Northwestern University. And she's the person who both founded the Oncofertility Consortium in the United States, but also, and importantly, built a collaborative infrastructure for bench-to-bedside fertility preservation research and clinical practice. And finally, I'll finish up by acknowledging my wife, who's also the lab manager and office manager. And she's the one who makes sure that I show up where I'm supposed to be when I'm supposed to be there. And she's a scientist, administrator, travel agent, sounding board, and many other things to me. So now that you know the players who were formative and sort of the role that they had in my development, I'll start to tell you the research story or the research and clinical story, and you'll begin to see how those different influences formed the direction of my research career. So for the people here today, most of you know about spermatogenesis, and I think you will also all know that spermatogenesis is a stem cell-based system where the foundational stem cell is a spermatogonial stem cell, or SSC, and this cell is defined by its ability to self-renew and maintain the stem cell pool, and to balance these self-renewing divisions with differentiating divisions that give rise to progenitor spermatogonia. These are diploid cells that undergo some number of transit-amplifying mitotic divisions before finally giving rise to primary spermatocytes that undergo 2 rounds of meiotic division that reduce to the haploid genome, and then the process of spermiogenesis that produces the terminally differentiated sperm that carries the haploid genome from dad and has a tail so that it can swim through the female reproductive tract and fertilize the egg. Well, all of this process of spermatogenesis happens here inside the seminiferous tubules of the testis, where every one of these seminiferous tubules both begins and ends in this structure here called the reedy testis. This is important because we're going to come back and visit this structure a couple different times during the presentation today. So sperm are made in the tubules, they flow out to the reedy testis and then via the efferent ducts into the head of the epididymis where they become more mature and learn how to swim. Now, the reason that I've emphasized the reedy testis is because this is the structure that we target when we want to infuse stem cells or other therapeutics into the testis. And the reason we do that is because by infusing into this space here, we can simultaneously fill all seminiferous tubules of the testis at the same time. You all know that the seminiferous epithelium is a highly organized epithelium with the stem and progenitor cells all located down here on the basement membrane. And then as the cells enter meiosis, they move progressively away from the basement membrane, undergo spermiogenesis, and then dump the mature sperm into the lumen of the tubule. Well, I told you about the pioneering work of Ralph Brinster, and in 1994, he described this method for transplanting stem cells into the testes of mice. And the way those initial experiments worked is that testes were obtained from mice that expressed the LAC-Z transgene, so all the cells in their body, including all of their testis cells, made this blue color. Um, if this tissue can be digested with enzymes to produce a single cell suspension and then injected into the testes of an infertile recipient animal. And then, if you recover those testes several months later, you can clearly see donor-derived colonies of spermatogenesis because segments of seminiferous tubule fill up with the blue color, which indicates the engraftment of a stem cell and its subsequent spermatogenesis that arises from that stem cell and begins to fill up segments of tubule. And if you produce sufficient levels of donor spermatogenesis in this recipient mouse, you can restore his fertility. So if you breed him to a fertile female, he can produce babies. And depending on how you design the experiment, some or all of those babies will have the donor genotype and phenotype, which is indicated here by the blue color. Well, I'm going to show you actual data of this, and just to tell you that in the actual data, the donor animal was hemizygous for the LAC-Z transgene. That means he had LAC-Z on one chromosome and not the other. So you would expect that about half of the recipients have the blue color, but the recipient in this experiment is one who's genetically infertile and incapable of producing sperm. So we know that all of the babies came from the donor-derived transplanted stem cells. Okay, so this is a story about two mice. On the left-hand side, you see a normal C57 black 6 mouse. You'll have to trust me that his testis is the normal size, but when you do the histology, you'll see that it looked like the cartoon that I showed you with multiple layers of germ cells in the seminiferous epithelium and a lumen where you can see that sperm have been deposited. Now, the recipient mouse in this case is called a W mouse. It has a mutation in the c-KIT receptor tyrosine kinase that means that it cannot produce spermatogenesis from stem cells. And I think you can see the stark contrast in the seminiferous tubules here because they're almost entirely empty. They can contain few, if any, germ cells. So the question that was asked by Takashi Shinohara, who is the lead researcher on this, is would it be possible to obtain stem cells from the testes of a wild-type animal and transplant them into the testes of this infertile animal where they would regenerate spermatogenesis? Of course, there's an assumption here that although the germ cells are missing, all the other cells in the testis, the somatic environment, are working appropriately and would be able to support spermatogenesis from the transplanted cells. So I'm going to show you how this transplant works. So for a mouse transplant, we access the testis by making an incision in the abdomen. And here we pulled the testis out on top of the belly of the animal, and we're looking down with a dissecting scope. Now in the mouse, the important structure, that reedy testis, is located right here on the outside of the testis so that we can see it visually. These is a bundle of efferent ducts, and we're simply inserting a pulled glass capillary pipette till the tip comes out into the reedy testis space. And then when you apply pressure, you can see that all of the tubules of the testis fill up at the same time. This is what a testis should look like when it's done. You don't want to overfill the testis because if you do, it'll clamp down on these blood vessels, and actually there will be no testis when you come back a couple of months later. So it's important that you don't get an ischemic event It's important to fill up just enough, but not too much. Well, here's the result of that experiment. Remember, we transplanted cells that came from an animal that expressed the LAC-Z transgene. And so at 2 months after transplantation, you can clearly see donor spermatogenesis. Cells were transplanted either into a pup recipient, this is a 5 to 8 day old pup, or into an adult recipient. And one of the things that you can see immediately is that the efficiency of engraftment and regeneration of spermatogenesis in a young testis is way better than it is in an adult testis. They both work, but it's way more robust in the young testis. And if we look at 5.5 months, you can see the result of the young recipient is quite dramatic because the testis has grown almost to the normal size. And if you look at the histology of that testis, you remember the recipient animal had completely empty tubules before. If you look at the histology now, you can see that almost every tubule is filled up with normal-looking spermatogenesis. In contrast, although that does happen in the adult, you can see the adult still has lots of empty tubules. So again, the point is the young recipient environment is much more supportive of donor engraftment and spermatogenesis than the adult environment. And the really important endpoint here is that normal fertility was recovered in 3 out of the 3 pup recipients, but 0 out of the 6 adult recipients. So in the adult, you could regenerate spermatogenesis, but not enough of it to restore normal fertility. And indeed, when you bred those animals, they were able to produce offspring. Here I'm showing actually fetuses. And like I said, about half of the fetuses have the blue color, but we know these all came from the donor cells because the recipient was genetically infertile and completely incapable to produce sperm. So when I came to Pittsburgh, this technology had already been or was in the process of being translated to many other animal species. That includes mice, rats, pigs, goats, sheep, cows. So it's a very robust technology that was translating across all species, and we thought that there might be application in the human clinic in the future. And of course, you all know that our focus has been on people who are receiving medical treatments for cancer or other conditions that can cause them to be permanently infertile. Adult patients, of course, have the option to preserve their future fertility by cryopreserving eggs or sperm or embryos prior to their gonadotoxic treatment, because we know that those cells can be thawed in the future and used for reproduction. But unfortunately, those options are not available to prepubertal boys and girls simply because they're not yet producing mature eggs or sperm. And we think this is an important human health concern because the really good news is that the 5-year survival rate for kids with cancer is now over 85%, which means that we, due to improved therapies, are producing a lot of survivors who still have their entire reproductive life in front of them. And these kids, of course, aren't thinking about the family that they might want to have in the future, but maybe that's something that we as a scientific and medical community need to help them to think about. And of course, they need to think about it prior to initiating gonadotoxic treatment. Well, as it pertains to the boys, even though they don't have sperm in their testes, they do have the spermatogonial stem cells. Those are present from the time of birth. And so there's the potential that we could preserve those cells and that they may have the potential to regenerate spermatogenesis using the method that I just showed you that was developed in 1994 in mice. And because we believe there will be future technologies that are in the research pipeline and might emerge one day in the clinic, there are centers worldwide that are actively cryopreserving testicular tissues for boys, also ovarian tissues for girls, because we believe in the future we'll be able to mature those tissues and produce eggs or sperm that can be used in reproduction. Looks like I lost a slide there, I think. We'll see what happens as we go along. Anyway, so when I came to Pittsburgh, like I said, I was recruited here by somebody who was an expert in using the non-human primate model, and specifically an expert in assisted reproduction in non-human primates. That was Jerry Shatton. And because of his support and because he gave me my first 20 male monkeys, Brian Herman, who was a postdoc in the lab, was able to use that as a monkey model of cancer survivorship. So what Brian did is he took a biopsy from the testes of these monkeys. Because we were planning to do a stem cell transplant, this tissue was digested with enzymes to produce a single cell suspension that was then cryopreserved. The animals were then treated with chemotherapy to destroy their spermatogenesis. And after their sperm counts went to 0, their cryopreserved stem cells were transplanted back into their testes. Before we did that, we treated with a lentivirus that carried the GFP gene. So if the ejaculated sperm contained the lentivirus or the GFP gene, we know that that came from the frozen, thawed, and transplanted cells. So the next question was, how do you inject cells into a monkey testis and eventually into the human testis? Well, it turns out that the reedy testis is not present on the outside of the testis like I showed you for the mouse, but rather it's here in the center of the testis. So here you can see the reedy testis. It's an echodense white line. All the seminiferous tubules are up here and down here. There's no point in accessing this surgically because you won't be able to see the reedy testis, but we can see it on ultrasound. And so we're able to insert a 25 gauge needle through the base of the scrotum into the reedy testis space. There's microbubbles in the cell suspension. So when we infuse, you can see first that the reedy testis is filled from end to end, and then eventually that cell suspension moves up into the seminiferous tubules of the testis. Because we can see all this on the ultrasound, we know for sure when the needle's in the correct spot because we can see the cells go where we expect them to go. So here's an example of one monkey who got an injection of his own cryopreserved cells. You can see that he was producing sperm, but after the chemotherapy treatment, his sperm counts went to 0. So at that time, we thawed out his cryopreserved testicular cells and transplanted them back into his testes. And about 30 weeks later, you can see that we began to see sperm in the ejaculate. It rose up basically into the normal range. And we know that at least some of those sperm contained the donor, were from donor cells that had been cryopreserved and transplanted, because when we did PCR for the lentivirus and GFP gene, we could see that it shows up in the ejaculate at about the same time that sperm showed up. And of course, that marker wasn't present prior to transplantation. We did go on to test the function of these sperm. We did that by sending them to our collaborators at the Oregon National Primate Research Center, who have a monkey IVF laboratory, and we found that the sperm were capable to fertilize monkey oocytes, leading to pre-implantation embryo development up to the blastocyst stage. We haven't yet produced a baby using those embryos, so that's something that still remains to be done so that we can document that healthy progeny can be produced from this experimental technique. All right, so based on what I've shown you, both work done by us in monkeys, but also by others in many other species, we believe that spermatogonial stem cell transplantation is a mature technology that may be available in the clinic in the future. And because we believed that, and the data that I just showed you in the monkey we produced in around the time of 2010 to 2012 or so, we began asking the question, is it reasonable to begin cryopreserving testicular tissues for patients because we think we're going to have a fertility restoration technology available for them in the future? So for us, the next step to the clinic was to establish our fertility preservation program. That happened in 2010. It was during that year that we received regulatory approval to begin cryopreserving both testicular tissues and ovarian tissues. And since that time, we froze our first tissues in 2011. And since that time, we've cryopreserved testicular tissues for 1,159 boys and 709 girls or women. I'll tell you that these numbers are about 1 week out of date, and we cryopreserve so many tissues these days that you can probably add about 5 or 6 or 7 to each one of those numbers because these are last week's data. Something really funky happened on this slide. I presented this slide so many times that I can tell you what it says. This is a pie chart that tells you about the medical conditions that patients have that come to us to cryopreserve their tissues. And the majority of diagnoses that we see in young patients are represented by the largest portions of this pie chart, and I'll try to remember what they are, but the most common ones are sarcomas, which I think is this one, leukemias, lymphomas, which is this one, and then neural cancers, which is indicated by this one. The big black pie-shaped wedge, which is about 30% of our patients, these are actually patients who are getting bone marrow transplantation for non-malignant conditions like sickle cell disease, beta thalassemia, and others. And finally, a growing population of patients for us are indicated by this purple wedge of the pie chart, which are patients with gender dysphoria that are on or getting ready to start gender-affirming treatments. I have to tell you that due to the political climate in the U.S. today, this program is shut down for the time being. So we provide the support the best that we can for the transgender community, but we cannot perform surgical procedures to cryopreserve tissues at this time. We're proud of the fact that patients have come to us from all over the world to access experimental fertility preservation options that are available in Pittsburgh, but you can imagine that need to travel can be a barrier in access to care, either because a family has a sick child that isn't able to travel, or because the cost of travel could be prohibitive. And so several years ago, we established a coordinated network of academic centers around the U.S. that now numbers over 50 sites. And under this mechanism, patients can have surgery to collect their ovarian or testicular tissues wherever they're being treated for their primary disease. And rather than the patients traveling to Pittsburgh, it's just their tissues that travel to Pittsburgh, where we have the expertise and the infrastructure to process and to cryopreserve those tissues. When we receive tissues in Pittsburgh, regardless of whether it's one of our own patients or from one of the coordinating centers, the processing looks like this. If it's testicular tissues, the tissues are simply cut up into small pieces that measure between 2 and 5 millimeters across. If it's ovarian tissues, we dissect the cortex off of the ovary, because the ovarian cortex is where the immature primordial follicles reside, and we cut those tissues into strips that measure 0.5 by 2 centimeters in size. And finally, for either ovarian or testicular tissue, we use computer-controlled slow-rate freezing to cryopreserve the tissues. And for all of those patients that are research patients, which is most of them, 75% of the tissue is cryopreserved for the patient's future use, and 25% is allocated to research. We're very grateful for the contributions and the participation of our patients in research, because it allows us to use patient data or tissues to understand the impact of their disease or medical treatment on their gonadal tissues in the process of spermatogenesis or oogenesis, and also to develop next generation reproductive technologies or reproductive options that we hope will be available for patients in the future. And so with that, I think that's my last slide. So I'll pass the baton to Amanda, who will tell you about some of the research that we do with patients that is and will allow us to provide additional reproductive options to our patients who are seeking fertility preservation care. Amanda?
Okay, I believe you can hear me, and you can also see my screen now. So first, I would like to thank you for the opportunity to speak here today. I could have probably gone before Kyle, because that's always a feat to follow him. He's such a great presenter. And so what I'm going to do here today is I'm going to talk about a portion of our research that we've learned from our patients that we've treated at the Fertility Preservation Program through our Center of Reproduction of Transplantation at UPMC McGee. More specifically, talking about searching for sperm results and their outcomes for patients that are peripubertal or adult. I'm going to move to the next slide. Okay, so we know that there are fertility preservation options for boys and men that are going to face gonadotoxic treatments. For adults, of course, they can cryopreserve sperm, and that then could be later used for in vitro fertilization or ITSE. For prepubertal individuals, we only have the testicular tissue cell cryopreservation that Kyle had mentioned previously, and the hopes are that once that is frozen, that can then be transplanted back later, either through cell suspension or through grafting. And that can also eventually lead out to ability to have children in the future. And really, we're focusing here on our peripubertal individuals that may not be able to save a semen ejaculate cryopreservation, but are able to, are going to have testicular tissue cryopreservation, and there may be sperm that is in their testis. And so we began just a little bit more about our center. We have started cryopreserving testicular tissues since 2011. And this was really just mostly at UPMC alone, was from 2011 to 2015. In 2015, we opened up our center to receiving tissues from coordinating centers under a research protocol. So we were continuing to cryopreserve for more. We opened up our center in 2019, and we also started our transgender, gender affirming care cryopreservation in 2019. And so, as Kyle mentioned, we actually, as of yesterday, have 1,163 patients that we have cryopreserved tissue for. And of those, since 2021, we have initiated a way that we wanted to offer the ability for us to search for sperm in testicular tissue of individuals to potentially be able to have to know whether or not there is sperm in that testicular tissue. And so we have this is a timeline of our history of searching for sperm in peripubertal and adult testicular tissues. So we started in May of 2021, and we piloted searching for sperm in tissues from our local patients. And so this was a small sub cohort before we kind of expanded this to being offered to our coordinating centers. And in that case, we had a primary cutoff of the individuals needed to be older than 10 years old, and we searched for those, and we learned information as we proceeded that actually we're not really finding any sperm in children that are under 12 years of age. So this data that I'm going to present here is actually going to represent data that comes from searches that we had a total of 104 between May 2021 and today. And one thing to note here is that although we started this endeavor in May of 2021, we had several different things where we expanded our opportunities here. So first, this was local in May of 2021. We offered this pilot then to research coordinating centers in April of 2022, so that expanded our numbers. In July of 2023, we offered this for searching for sperm in contracted services agreements, and then we updated in 2024 our coordinating center research protocols to provide consents and other things to help educate about this potential opportunity. And so with that, really from July of 2023, that's where we have had most of our patients that we're looking at from here. So if we had between May and now, May of 2021 and now, we had 229 peripubertal or adult patients. And of those, we had 104 that consented to search for sperm. So that's about 45% of individuals that chose to have us look for sperm in their testicular tissue. This was not offered to everybody throughout this timeline, so probably more of an acceptance rate would be referring to the individuals that had cryopreserved tissues from July of 2023 onward. Of that, we have 94 peripubertal adult TTCs that have occurred, and of those, 70 or 74% consented to search for sperm. Okay, and so over those 104 attempts, we have found 20, or we have found sperm in 20 of those tissues. And in that case, that's about 19.23% of successful searches for sperm. Now, I've broken down this data into a couple of different attributes that we can see. So the first that I'm showing here is the differences in diagnoses and the rates of success. And so you can see here we have a large population of gender dysphoria patients. These are all patients that have been seen here. And we have oncological and non-malignant blood disorders that represent a large portion of our population that Kyle had mentioned in his diagnosis charts. We have success that is of successful searches for each of those different types of diagnoses types. The ones that we have not had success for is some of our disorders of sexual development, such as Klinefelter syndrome, represented here in this population. But overall, it seems that we do not have that diagnosis is probably not the main reason on whether or not sperm can be found. But we more than likely it's going to be related to age and more specifically Tanner stage or prior treatments to before cryopreservation that would really affect the likelihood or outcome of here. Of note, it does look like the gender dysphoria patients are only about 10% of the time. This, I will say that most of our gender dysphoria patients are on the younger side. So there's probably an age confounding factor that is affected here. But I'll break that down in the upcoming slides. And so if we look here, this is breaking this out by age. So you can see our youngest individuals were 10 years old to 29. These individuals that are in their 20s, most of them are gender dysphoria patients. So they have had puberty blockers and or estrogen. So the fact that we have not found sperm in here might be related to prior treatment and not in age. But you can see here that basically after the age of 12, we have about a 21 to 30% chance of finding sperm in individuals. More specifically than just age, if we know Tanner staging, that can be a little bit more of a predictor of success rate. So here we have this broken out by Tanner stage. So if it's 1 or 2, we have had 11.1% success rate. If we know that the Tanner stage is 3 or higher, we have a 47.6% success rate. And if we don't, Tanner stage is unknown, then we have about a 12.2% success rate. This, of course, has then a mix of prior treatments and other types of things that may affect this as well. And so then if we break these down into different cohorts, you can look at diagnosis and treatment and how that may affect. And so here I'm going to first represent our oncological diagnosis and prior treatments. So these would be our leukemias, lymphomas, sarcomas. And we have here represented that we have 43 patients that represent this category. And of that, we have had success in finding sperm. And across different categories, we have different success rates. So on average, we're about 30%. And that can be if we have no prior treatment for testicular cancer or regular cancer, you have about 26 to 40% success rate. We do have, our numbers are increasing now that we have some information related to prior alkylating chemos. If they are less than 4 grams per meter squared, there's still about a 40% success rate in finding sperm. Our other different types of treatments, we are collecting or continuing to collect data on these, but they are relatively low numbers, so we can't necessarily output on what those success rates are. But as you can note, all of these colors here represent the different types of prior treatment. And you can see that they are represented on both lines, either sperm was found or not. And this also separates it out by age. So age is a big indicator, but then also prior treatment is as well. Next, we'll talk about our non-malignant blood disorders and prior treatments. And so these are our sickle cell disease, our beta thalassemia patients that are going to have bone marrow transplant, and that's why they're undergoing testicular tissue cryopreservation. And so with this patient population, they do have a lot of prior treatments, including blood transfusions and hydroxyurea, which may contribute to some infertility as well. And so here we have about 14 patients that are represented in combination of whether or not they had just blood transfusions, hydroxyurea, or a combination of both. We are finding some success in this patient population as well. Again, our numbers here are relatively low, so we'll be excited to report this as we gather more outcomes. And our last cohort that I want to talk about is our gender dysphoria diagnosis patients and their prior treatments. And so this is our largest population that we have represented here in our search for sperm outcomes. In this case, we have several observations that we can report. So you can see here that we have had success in finding sperm in our gender dysphoria patients versus not finding sperm. This all relates down to different types of the treatments that they have been on. So in the no treatment category, most of these individuals, you can see the green dots here, are younger because they have, are waiting to go on to treatment after the cryopreservation. And so likely the no treatment, the age impact on the no treatment group is really what you're seeing, the reduction in finding sperm. But anyway, we have a success rate of 11% for those individuals. Individuals that are on testosterone blockers or estrogen for over 6 months, we have not found any sperm in those individuals despite their ages. So you can see here the purple is represented by those individuals. However, we have had one success in an individual that had testosterone blockers and estrogen for less than 6 months. As well as just the testosterone blockers. These numbers are relatively low still, but we can probably say for sure that individuals that have had testosterone blockers or estrogen treatment for over 6 months, we're likely not going to find sperm in their testicular tissue. And so we really also, one of our other questions that we wanted to ask was whether or not sperm can be recovered after transport. So as we mentioned before, we have a coordinating centers mechanism. Many sites will send us their tissues to cryopreserve. This is going to be transported at 4 degrees to us and usually overnight. And we wanted to know whether or not we would be able to find sperm after the storage or what effects that might be. So we did a side study with mice where we took their testis and we separated it out and mimicked a transport where we had it stored at 4 degrees at room temperature or at 37 degrees overnight. And then we did a comparison where we looked at different semen parameters. And so in this case, representing here, we have our 1 hour data points and our 24-hour data points in gray. And you can appreciate that there's no difference even across, no significant difference across the temperatures for sperm count or morphology. However, sort of our more qualitative types of functional assays related to sperm viability or sperm motility, those do decline at over 24 hours if they are at 25 degrees or 37 degrees. However, there is still a retention of that at 4 degrees. And so this was hopeful to us that we would be able to find that, and that likely it can result in some type of functional ability. And so we moved this to when we looking at our human data, I can break down the cold incubation time of testicular tissue. And so this represents the length of time from surgery to cryopreservation, and whether or not we were successful in finding sperm. And you can see that 0 to 6 hours, these are going to represent our local cases. And the 12 to 48 hours, this is going to represent our cases that are coming from across the U.S. And there doesn't appear to be, we're finding success rates across all of those incubation times. You know, to note that these 6 categories that is broken down, this represents a mixture of ages, prior treatments, and diagnosis, which may also contribute to those success rates. And also, as a caveat here, it's unknown whether or not this cold incubation time affects the actual function of the sperm. I'm just representing that we are able to find that sperm when we go to cryopreserve. And so with that, our conclusions from our search for sperm outcomes is that approximately 20% of the time, we are finding sperm in peripubertal and adult testicular tissues when we go to cryopreserve. There has been no sperm that's found in individuals that have been on puberty blockers or estrogen treatment for greater than 6 months. Promisingly enough, we are able to find sperm in testicular tissues that are transported at cold temperature. And we're going to continue to update and learn from our search for sperm experiences to better counsel patients in the future. And with all of that, I want to recognize our current Center of Reproduction and Transplantation, our laboratory supervisor, andrology specialists, etc. They do a lot of our tissue cryopreservations and these searches for sperm. So really this data that is presented here is a reflection of their clinical work, and this represents our current andrology specialists, but there have been past ones that have helped as well. And so with that, I want to thank you all for your attention.
Okay, thank you for those 2 great presentations. We will now go over to the Q&A session. We already have some questions in the question box, but if any others pop up, you can still send them in. The first question was for Professor Orwig, and it was, would it be possible to place fragments of tissue back in men instead of only cells?
Yes, so the answer to that is yes. And in fact, we are approved both to do stem cell transplant and testicular tissue transplant into human patients. The reason that I told you only about the cell transplant today is because when we had the success doing that, that's what prompted us to begin cryopreserving tissues for patients. But we continued to do work in the research laboratory and later published that we could produce a live and healthy monkey offspring from testicular tissue that were transplanted under the skin. Those tissues were recovered, sperm were released, and we used to fertilize eggs by ICSI and transferred the embryos to produce offspring. In fact, we'd done our first stem cell transplant in a human patient about 1.5 years ago, so we're following that patient to see whether they ever produce sperm in their ejaculate. We haven't seen that yet, but we do see round cells in the ejaculate, and at least the molecular analysis indicate that we may see some differentiating germ cells, but again, not yet sperm. Ellen Goosen's group in Belgium did the first transplant of intact pieces of testicular tissue into patients just this past spring. She transplanted both under the skin and also within the testicular tissue. And so we're all eagerly awaiting to see whether those tissues mature when they recover them, and whether they're able to recover sperm. This is becoming a long answer, but I will say, if I had a bias and I had nice intact pieces of testicular tissue, I probably would opt for the testicular tissue transplant over the cell transplant, but I won't spend the time going into all the reasons for that. But just to tell you that probably would be my preference, and also how I would counsel patients.
Yeah, interesting. Thank you, Kyle, and also Amanda. I now have a question for Amanda. This is about what the sperm was used for. So the question is, what method was used to search for sperm in peripubertal tissue, and was the sperm frozen for future fertility treatments?
Okay, yes, thank you. So to answer that question, our tissue, basically we did a similar procedure as we would for a processing a microtessie or a tessie sample. So basically we shred apart the tissue using like a needle, or in our case, it's like stab knives, but essentially we're separating that out. And then we're looking at that under the scope. And then there was a follow-up portion to that question as well. I think that was whether or not, if we found it, if we cryopreserved.
And
So
how do you cryopreserve?
Yes. So we actually cryopreserve depending on the results, or how we cryopreserve depends on the results, but we ultimately cryopreserve that portion that we have searched for. So if we find sperm, we cryopreserve using just like a Regio freezing media, sperm media, follow our regular protocol that we do for freezing sperm. And then if we do not find sperm, then we will centrifuge down the cells, and then we will do the slow freezing process that we do for testicular tissue cryopreservation. In that case, it's not going to have the intact tissue that we would normally do for like tissue grafting or other types of things, but there are still cells there that are important that could potentially be used in the future. So depending on the outcome, we'll freeze it one of two ways. But the big result is just knowing whether or not there's sperm there that could potentially be used.
Thank you.
And just to add a little bit to that, is just a portion of the tissue that's dissected to see if there's sperm in there. If we find sperm, we freeze it like you would freeze a tessie sample, or like we would freeze a tessie sample, which is in liquid nitrogen vapor, followed by plunging in liquid nitrogen, which of course is not the way that we freeze intact pieces of testicular tissue. That's the computer-controlled slow freezing.
No, so not all tissue is sacrificed for
Mhm.
searching for sperm in these patients.
Yeah.
That's correct. And the other thing is, this is experiments that we'll do in the future, but I have a feeling that the sperm that are in the tissue that's frozen by computer-controlled slow rate freezing are probably fine. So we opt to use the method that most people would use in an andrology lab to freeze testicular tissue with sperm. But it could be that the sperm that's in the intact pieces of tissue that are slow frozen are probably just as good. Of course, that'll become a mouse experiment where we can fertilize and show that we can produce healthy offsprings from it.
Then another question, and I think it's probably most for Professor Orwig. Aren't you afraid of relapse of the cancer?
Yeah. So, of course we are, and here you're talking specifically about people with testicular cancer. Of course, people with leukemias or lymphomas could have malignant cells in their tissues, and in a small percentage of cases, people with neuroblastoma may have malignant cells in their testicular tissues as well. And so for those patients, we need to be concerned about the potential of reintroducing a cancer. And so when we counsel the patient, we will tell them that although the transplant technologies that we talked about may be the more mature ones, they may not be appropriate in that circumstance, because we wouldn't want to reintroduce a cancer. Therefore, we're relying on methods to mature those tissues outside the body and produce eggs or sperm. Those technologies are earlier stage. I don't think they're quite ready for deployment in the clinic. And so it's a gamble for the patient. They have to weigh the risk and the benefits, but here the benefit is gambling on a technology that's early stage and that we don't know whether it will ever be available in the clinic.
Thank you for this answer. Now to Amanda again. This is a question regarding the transgender testicular tissue. So you were not able to find any sperm cells in those patients that have been on testosterone blockers and estrogen after 6 months of treatment. So do you see a difference between both tissues in terms of number of germ cells? So did you, for example, check the histology or the morphology of this tissue?
Yeah, so that's a excellent question. I don't necessarily have the data here to fully present, but we have looked histologically, and we also have done a study where we have looked at that functionally as well, by doing transplants into mice, and both despite treatments functionally, they are able to have stem cells colonize the mouse testis, indicating that there is functional spermatogonia still there. Histologically, I mean, you still see the beginning stage or the spermatogonia. You're just not seeing sort of the full expansion into spermatids.
No,
Yeah.
no, clear.
Yeah.
Clear.
Okay, then again, a question for Professor Orwig. It's also a question that I had because in your presentation in the beginning, in mice, you said that the young recipient testes are better to restore spermatogenesis compared to adults. And then the question is, do you think that if we extrapolate this into human, that stem cell transplantation into adult recipients will result in a restored capacity to have children without the use of IVF?
I'm going to answer the last part first. And actually, by the way, so I think that regardless of whether you do a stem cell transplant or testicular tissue grafting, IVF is going to be necessary. In order to restore fertility in mice, you have to go to extraordinary measures that usually involves, first of all, an ideal recipient who has nothing in their seminiferous tubules, so it's a great place to transplant. Secondly, we pool cells from multiple donor mice together, and then we use some mechanism to isolate and purify the stem cells before we transplant. So it's really ideal circumstance. In the human patient, we take a small biopsy from a boy when he's young, and just a small portion of one testis. And so it's reasonable to expect that there are only a few stem cells there, and that when we transplant, they're going to produce small pockets of spermatogenesis in the testis. But maybe even not enough for any to emerge in the ejaculate, and definitely not enough to restore normal fertility. So the answer to the last part of the question is that I think IVF or assisted reproduction is the path, no matter whether you do stem cell transplant or tissue transplant. Regarding the transplant into young patients, so a 5 to 8 day old mouse is equivalent to a pubertal boy. And I believe that puberty would be a great time to transplant because during that time, the testis is growing, the gonadotropic hormones are being produced. And so it's creating a lot of new niches for the stem cells, not only to graft, but proliferate and occupy the next niche and the next niche, et cetera. This is actually, but we don't transplant during puberty. And the reason that we don't do that is really influenced a lot by discussions we had with the ORCHID-NET collaborative, where people asked this question: why would you do an experimental transplant in somebody who's so young that you don't even know yet whether they're infertile or not? And this is a reasonable question. I think it makes perfect sense to get a semen sample from the patient and find out if they're actually infertile, because if they're producing sperm, there's no need to do an experimental stem cell transplant, which of course has some risk associated with it. And so because of that, the youngest age for our stem cell transplant is 16 years old. We don't require a semen analysis because there are some people that for religious reasons or otherwise won't do it, but we heavily recommend getting a semen sample done. And of course, if there were sperm there, we would recommend not to proceed with a stem cell transplant. Final thing, although we don't do this at pubertal age, my very strong preference is to transplant as early as possible, because it may take a long time to produce enough spermatogenesis that the doctor can go then retrieve sperm from the testes. And it would be good if the transplant was done way in advance of the time that the patient actually wanted to start their family, because of the amount of time it'll take to produce spermatogenesis.
Thank you. There's also a question about logistics. So when you transport this tissue throughout the US, how long can you keep it safe? Because there are quite some challenges with participants collaborating with fertility center.
I think Amanda can tell us on average what the time is.
Yeah, so on average, most of our tissues come to us within 20 hours between removal of surgery, travel, transport, and us starting to process. We have had cases where that has been delayed out to, I believe, 72 or 96 hours. Kyle, do you remember?
I think 96 hours was the longest one. It's kind of a it's an unfortunate but funny story, but represents real life. That is that a site had done the surgery to collect tissue on a Friday, but never told us about it. And on Monday, they said, hey, we have this tissue in the refrigerator. Can we still send it? And, by that time, surgery's been done. So what choice do you have? So they sent it and we received it on Tuesday. So it's 96 hours from the time of surgery. Of course, we cryopreserve the tissue because the patient's already undergone the risk of a surgical procedure, but we can't comment on the quality of the tissue except to comment on whether there were still germ cells present in there or not.
And in that case, there were germ cells that were still present, but again, you don't know necessarily the function of it. And then to say with that, obviously, the sooner that you can process, the better. But we have results from patients that have been around the 20-hour mark.
Thank you.
Then we still have a lot of questions. Then another question for Professor Orwig. Do you consider using sperm even when patients have previously received gonadotoxic treatments such as alkylating agents? And do you have a time frame between the preservation of testicular tissue and the last dose of chemotherapy received?
Yeah, and this question might be prompted by something that Amanda showed in her data, which is patients who had had a previous chemotherapy before they cryopreserved, or before we did the search for sperm. And the answer is, in that circumstance, I would treat it the same way that you would treat an adult patient who had had a recent exposure. By the way, every single one of our patients who's doing fertility preservation is counseled that we don't know the impact either of the freezing or of previous medical treatments on the quality of their sperm or the risks for passing a birth defect on to children, and the patients need to sign that they understand that. But I think the advice in an adult clinic, and others can educate if this isn't true, but the advice in the adult clinic typically would be to wait 6 months to 2 years before trying to preserve a semen sample or trying to have a baby after your last exposure to a chemotherapy. So I think the same thing when you do a search testicular tissue for sperm, the guidance should be the same. Is if it's more than 6 months to 2 years after the treatment, we would still have them sign a waiver, but that's probably safer than if the treatment was closer in time to when the sperm are collected.
Clear. There's just to clarify, there's one question that asks that whether all SSC transplant would eventually resolve in the need for a TESA or a micro-TC. What do you think?
I think that that's the reasonable expectation, that because of the small number of stem cells that I would expect to get from a young patient, that these would produce just small pockets of spermatogenesis, and the surgeon would ultimately have to go in and find those pockets and collect sperm from dissected tubules. You could imagine a future which may or may not ever come to fruition, but something that you can do in mice today, and that is that if we could figure out how to expand human spermatogonial stem cells in culture, then we can take that small number of stem cells and expand them to thousands or millions before you do the transplant, and then perhaps we would have a different outcome. Lots of labs around the world have reported on human spermatogonial stem cell culture, but I don't think there's really a consensus that there's any one method that actually leads to expansion. Although this is a major focus of what we do, it's possible that we'll never achieve that endpoint. So try to be very realistic when we counsel patients.
Very clear.
We still have a question, I think mostly for Amanda, because it's again on transgender patients. The question, if estrogen treatment is stopped in transgender patients, could spermatogenesis recover? And if yes, do you have an idea on how long it needs to be stopped before you can go for cryopreservation?
Yeah, so that's a good question. I don't necessarily have the full data to support this, but I know anecdotally that we have patients that have come in, stopped their estrogen treatment, and have been able to save a semen do a semen cryopreservation within 3 to 6 months after stopping their estrogen treatment. The numbers there are relatively low, infrequent, and not something that I'm going to report out as official. But anecdotally, it seems like if you stop estrogen treatment and you wait enough time, that there can be the return of spermatogenesis.
And there's limited, if any, data if those sperm that have been recently exposed to gender-affirming treatments have normal function and can produce healthy babies. So that it'll take a while to accumulate enough of these individual patient stories to begin to have a clear understanding of that.
Yeah. There's now just one question left, and it's actually going more into the future, because here's a participant that's quite worried about the chance of relapse, and she is asking about the possibility for in vitro maturation of the tissue. What's your standpoint on that? What's your idea?
I can start the response, and Amanda may have something to contribute to. But one of the things that we're just starting to look at is when we're processing the ovarian tissue, we're removing the medulla, we're actually maturing follicles reside. And so we're just starting a study now that Amanda will lead to see whether we can actually mature those follicles ex vivo and cryopreserve the follicles, and therefore there'd be no need to put the tissue back in. We do know that we can see some larger follicles, and we haven't yet started the IVM process, but there are other places in the world that are doing IVM from eggs that are released from ovaries, and it appears to be successful. I think there are even live birth outcomes, but I'm not going to quote them because this isn't our data, and I don't actually, on the top of my mind, I don't know. So that's something you could do at the time of ovarian tissue cryopreservation. That would be an ex vivo. Here's how to produce a mature egg. There are many groups around the world who are working on methods to mature ovarian or testicular tissue in vitro to produce eggs, and those things are very early stage. They're in lower animal models, and we have quite a ways, I think, to go before we see them emerge in the clinic. Although I will say that I believe it's Christine Wynn's group who showed that they can mature testicular tissue up to the point where they see spermatids, starting with a prepubertal tissue, and then after some time in culture, they can see spermatids. So there's hope.
Yeah, hope on both the transplantation and also perhaps on in vitro maturation of the tissue. Well, I think then this concludes our webinar. We're a bit over time. I'm very happy that a lot of people still joined until the end of the Q&A. And maybe Emily, some last words from the NYRA.
Well, we're also very thankful to have heard your backstory, let's say. It's always interesting for young researchers to get a bit more knowledge on how to roll in and how we can all start a career in academia. I think thank you for everybody to join, and I think we can close off here. And it would be nice to see you again in the future.
Yeah,
thank
thanks.
you. Guys. Fun.
Thank you.
Thank you.
Thank you. Thanks a lot.
Bye-bye.